Hello everyone. Today we are going to study about composition, structure and function of carbohydrates, part 1. What are carbohydrates? Carbohydrates are the most abundant biomolecules of the biological system. It contains carbon, hydrogen and oxygen. If you see the name, it is carbohydrate. The carbo stands for carbon. Hydrate stands for H2O. Clear? So carbohydrate consists of carbon, hydrogen and oxygen. Carbo means carbon, hydrate means H2O. So carbohydrate is a hydrate of carbon. What is its chemical formula? The chemical formula is C for carbo, H2O for hydrate. Whole N. Why N? Because carbohydrate contains more than one carbon and hydrate molecules. Hence, the N is used. Clear? So, carbohydrate means the hydrates of carbon. So, it contains carbon, hydrogen and oxygen. The chemical formula is CH2O whole N. Why N? Because it contains more than one carbon and hydrate molecule. Now, there are three types of carbohydrates. One is monosaccharide, oligosaccharide and polysaccharide. The word saccharide means sugar. Mono means one. So monosaccharides are the sugar that consists of one sugar molecule. Similarly, for oligosaccharide, saccharide means sugar. Oligo means 2 to 10 molecules. So oligosaccharides are the sugar compounds consisting of 2 to 10 molecules of sugar. Polysaccharide, again the saccharide means sugar. Poly means more than 10. When a sugar compound is made up of more than 10 molecules of sugar, then we categorize it as a polysaccharide. Clear? So carbohydrate is a hydrate of carbon. Hence it contains carbon, hydrogen and oxygen. Its chemical formula will be CH2O whole N. N because it contains more than one carbon and hydrate molecules. Carbohydrates can be divided into three categories: monosaccharides, oligosaccharides and polysaccharide. Saccharide word means sugar. Mono means one. So monosaccharides are the carbohydrates containing one sugar molecule. Oligosaccharide, again saccharide means sugar. Oligo means 2 to 10. So carbohydrates that contain 2 to 10 molecules of sugar considered under oligosaccharide. Polysaccharides, when carbohydrates are made up of more than 10 sugar molecule, then we can call it as a polysaccharides. Now monosaccharides. What are monosaccharides? Just now we learned it is made up of one sugar molecule. Now that sugar molecule should contain at least 3 carbon atoms. Monosaccharides means the sugar molecule, the carbohydrate that contain one sugar molecule, and that sugar molecule should have at least 3 carbon atoms. Now the simplest monosaccharides are CH2OH, C double bond O, CH2OH. This is dihydroxyacetone. And another one is glyceraldehyde. Both contains 3 carbon atom. Okay. Carbon number 1, carbon number 2 and carbon number 3. Here also carbon number 1, carbon number 2 and carbon number 3. So both the monosaccharides contain 3 carbon atom. Now what is the difference between both the monosaccharides? The functional group in dihydroxyacetone is a ketone group, whereas the functional group in case of glyceraldehyde is a aldehyde group. So dihydroxyacetone is a type of a monosaccharide which can be categorized under ketoses. Ketoses means those who contain ketone functional group. Glyceraldehyde can be categorized under aldoses. Aldoses are the monosaccharides that consist of aldehydic functional group. Clear? So, on the basis of functional group, we can categorize monosaccharides into two varieties. One variety is of aldoses and another variety is of ketoses. Aldoses, those who contain aldehyde functional group, that is CHO. Ketoses, those who contain ketone functional group, that is C double bond O. Clear? So monosaccharides can be categorized under two categories depending upon the presence of functional group. If the functional group is a ketone group, then it will be categorized under ketoses. If the functional group is a aldehydic group, then it will be categorized as aldoses. So dihydroxyacetone is a ketoses type of monosaccharide, whereas glyceraldehyde is an aldoses type of monosaccharide. Next difference we can find from here. When we are numbering the carbon atoms, in case of aldehyde functional group, the functional group is the first carbon. CHO is the carbon number 1. Whereas in case of ketone group, C double bond O, it is a second carbon. Clear? So aldehyde functional group is present at the first position. The carbon is the first carbon, and ketone functional group, the carbon is the second carbon. Clear? So now again we can define monosaccharide as, how we can define monosaccharide now? These are aldehyde or ketone derivative of straight chain polyhydroxy alcohols. Why polyhydroxy alcohols? Because it contains multiple OH group. In case of dihydroxyacetone, here also OH, here also OH. Clear? In case of aldoses, carbon number 3 also OH, in carbon number 2 also OH present. Clear? So, monosaccharides are aldehyde or ketone derivative of straight chain polyhydroxy alcohols. Clear? Monosaccharides can be divided into two categories, aldoses and ketoses. If the functional group is an aldehyde functional group, that is CHO, we can categorize it as aldoses. If the functional group is a ketone functional group, that is C double bond O, we can categorize it as a ketoses. Aldehyde functional group constitute carbon number 1, whereas ketone functional group constitute carbon number 2. Clear? One more way by which we can categorize monosaccharides, that is number of carbon atoms. Because in the beginning only we said monosaccharides contain at least 3 carbon atoms. So depending upon the number of carbon atom present in a monosaccharide, we can always categorize monosaccharides into various categories. Now these are the types of aldoses. Now let's see the first aldoses, that is glyceraldehyde. How many carbon atoms? 1, 2 and 3. So it is a type of aldotriose. Aldo from aldehyde. Triose, why? Because 3 carbon atoms. So aldotriose. Next, this is carbon 1, carbon number 2, carbon number 3, carbon number 4. Here also carbon number 1, 2, 3 and 4. So these are the types of aldotetroses because of 4 carbon atoms. Next, carbon 1, 2, 3, 4 and 5. So third is the aldopentoses. Next, carbon 1, 2, 3, 4, 5 and 6. So type of aldohexoses. Clear? So monosaccharides can be categorized also on the basis of number of carbon atoms present. Now, One thing you must have noticed in these structures, everywhere it is written D. D-glyceraldehyde, D-erythrose, D-threose, D-ribose, D-arabinose, D-glucose, D-mannose. So why D? What does it mean? Let's understand this with the structure of glucose only. Now this is the structure of D-glucose and L-glucose. Now pause the video and see the difference between D and the L-glucose. So what is the difference? The positioning of OH around carbon number 5. The D-glucose and L-glucose differs only in the positioning of the OH around carbon number 5. Okay, if the OH is present on the right hand side, we call it as a D-glucose. The full form is dextroglutatory. If OH group is present on the left hand side, then we call it as a L-glucose or levorotatory. Clear? Rest all is same. So these two are the non-superimposable mirror images. So D-glucose means when the OH group attached with the fifth carbon is present on the right hand side, we call it as a D-glucose. If it is present on the left hand side, we call it as a L-glucose. D stands for dextrorotatory, L stands for levorotatory. This is for hexoses. Now for pentoses or trioses or tetroses, how we will find L and D form? So in pentoses, we won't be having 6 carbon, right? We will be having 5 carbon. So in that case, you will be looking for carbon number 4. The OH present around carbon number 4. That means the criteria to look is the carbon atom or the OH which is farthest from this functional group. The OH which is farthest from the functional group. The positioning of this OH helps us to determine whether it is a D form or the L form. Clear? The OH which is farthest from the functional group. That OH is present on the right hand side or on the left hand side will determine whether it is a D form or L form. Clear? These rest OH, the third OH, the fourth OH, the second OH doesn't matter for the D and the L form. Which OH is important? The OH which is farthest from the functional group. If that OH is present on the right hand side, we call it as a D form. If that OH is present on the left hand side, then we will call it as a L form. Clear? And these D form and L form are non-superimposable mirror images which are also known as stereoisomers. How we will find how many stereoisomers are possible for a monosaccharide? Let's understand this again with the structure of glucose. So first, to find the number of possible stereoisomers, we should find the number of chiral carbon present. What is a chiral carbon? Carbon can form 4 bonds. So if carbon is bonded with all different atoms or group of atoms, then that carbon is known as a chiral carbon. If carbon is bonded with all the 4 different atoms or a group of atoms, then that carbon is known as chiral carbon. Now, for glucose, how many chiral carbon it contains? Total number of carbon? 6, right? Now we have to find number of chiral carbons. Carbon number 1 is not a chiral carbon. Why? Because it is double bonded with oxygen. Carbon number 6 is also not a chiral carbon because it is bonded with 2 hydrogen. So the remaining carbons will work as a chiral carbon. How? Now consider for C2. The second carbon bonded with hydrogen, bonded with OH, bonded with CHO and bonded with this whole group. So all the four bonds are with different atom or group of atom. So hence C2 or carbon number 2 is a chiral carbon. Now come to carbon number 3. H one end, OH another end. This whole lot, the third bond, this whole lot, the fourth. So carbon 3 is also a chiral carbon because this carbon is also bonded with all the four different groups of atom. Come to C4, bonded with H, bonded with OH, this below one group, this above one group. So C4 is also a chiral carbon and similarly C5 is also a chiral carbon. So total how many chiral carbons are present? 4. Clear? Understood how we can find chiral carbons? Now possible stereoisomers can be calculated using the formula 2 to the power n, where n is a number of chiral carbon. So here for glucose, the number of chiral carbon is 4. So how many stereoisomers are possible? 16. Clear? So this how we can find the number of possible stereoisomers for monosaccharides. Now, again from these structures, one more thing we can notice. And what is that? If we see this one and this one, what is the difference? The red part only. The placing of H and OH around a second carbon is only different in these two structures. Similarly, for glucose and mannose or glucose or galactose, also it is the same. Now, this is glucose. The first structure is of glucose. The second structure is of mannose. What is the difference? Only the placing of OH around carbon number 2 is different in both the structure. Rest everything are same. So these type of molecules are called epimers. So what are epimers? Epimers are the compounds or the molecules that differ only by configuration around one carbon atom. Those are called epimers. In case of glucose and mannose are differ only around carbon number 2, where for glucose, the OH attached with carbon number 2 is present on the right hand side. In mannose, the OH attached with the carbon number 2 is present on the left hand side. Clear? Next come to the deketoses. So this is carbon number 1, 2 and 3. So this is again trioses because it contains ketone, so ketotrioses. Similarly, 1, 2, 3 and 4, this is tetroses. Ketone group at second position, so ketotetroses. Similarly, this will be ketopentoses. And this is ketohexoses. Clear? Rest all are same. That is why D are written. That depends upon the OH group which is farthest from the functional group. If it is present on the right hand side, then it is D. If it is present on the left hand side, it is L. Now, why everywhere we are studying or why everywhere only D form is written, not the L form? Because in biological system, D form is a dominant form or D form is mostly present in a biological system, not the L form. That's why whenever we study carbohydrates, we study with respect to the D form only. Clear? Now come to the stereoisomers for ketoses. Ketoses have less chiral carbon compared to aldoses. Therefore, less stereoisomers are possible for ketoses compared to aldoses. Glucose, we already studied that carbon number 1 is not a chiral carbon. Carbon number 6 is also not a chiral carbon. But the rest 4 are all chiral carbon. But in case of fructose, pause the video and find out by yourself how many chiral centers are present for fructose. This first one is not a chiral center. The reason is same, C is bonded with 2 hydrogen. Sixth one is also not a chiral center because C is bonded with 2 hydrogen. Second one is also not a chiral center because C is bonded with oxygen by double bond. So how many left? Only 3. Both are hexoses. Fructose is also hexose. Glucose is also hexose. The difference is glucose is aldohexose and fructose is ketohexose. So both are hexoses, that means both contain 6 carbon atoms. But for aldohexose, the number of chiral centers are 4. But for ketohexose, the number of chiral centers are 3. So ketoses have 1 less chiral center as compared to aldoses. Clear? So how many stereoisomers will be possible for D-fructose then? 2 to the power 3. Whereas it was 2 to the power 4 for glucose. Clear? Now one question from my end for you all. You have to tell me how many stereoisomers are possible for ketopentose, ketohexose and ketoheptose. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.